Hello everyone. Today we are going to discussing about the topic that is activation of amino acid, what is charged tRNA, and also about the enzyme aminoacyl tRNA synthesis. Let's move on to the topic. Activation of amino acids or charging of tRNA is the first step in protein synthesis or translation. So both these steps are catalyzed by the enzyme called aminoacyl tRNA synthetases. Amino acid activation refers to the attachment of an amino acid to its respective transfer RNA, that is tRNA. And this whole reaction takes place in the cell cytosol, that is at the matrix of the cytoplasm. Let's see some details about the enzyme aminoacyl tRNA synthetase. Aminoacyl tRNA synthetases are the only molecules in biology that know the genetic code. That is, this enzyme can literally read the genetic code and can join with the tRNA with its specific amino acid. So, this enzyme is specific for an amino acid. And this enzyme has three-part active sites. They are amino acid binding site, ATP binding site, and tRNA binding site. In the recent report, we can see there are at least 23 different aminoacyl synthetases and each enzyme is specific for an amino acid. So as we can see, the first step in protein synthesis is amino acid activation. It is simply the formation of aminoacyl AMP. Here, the amino acid binds with ATP and form amino acid AMP with the release of two phosphates. Here you can see the amino acyl tRNA synthetase and there is a amino acid valine and then the ATP, and this enzyme joins this valine amino acid with ATP and forming amino acyl AMP with the release of two phosphates or pyrophosphates. Now this amino acid is activated, or we can say the amino acid bound to AMP is called activated amino acid. So this aminoacyl tRNA synthetase joins with the amino acid valine with the ATP and forms aminoacyl AMP with the release of two phosphates. And the next step is formation of charged tRNA. Here you can see an uncharged tRNA where the corresponding amino acid is not present. So the tRNA having corresponding amino acid is called charged tRNA. So the second step is the formation of charged tRNA. Here aminoacyl AMP enzyme binds with the tRNA and form amino acid plus tRNA. Here we can see the uncharged tRNA binds to the active site of the enzyme, and this enzyme transfers the amino acid from aminoacyl AMP and form aminoacyl tRNA. So this is the aminoacyl tRNA. So after forming the aminoacyl tRNA, here AMP is released and charged tRNA with the amino acid is also released. So the process of joining tRNA with specific amino acid is called tRNA charging. So once again, here the amino acid tRNA, first step is amino acid activation. So here amino acid tRNA synthesized joins with the amino acid valine with the ATP and form amino acyl AMP enzyme aminoacyl AMP with the release of two phosphates. And this and an uncharged tRNA binds to the active site of the enzyme, and this enzyme transfers this amino acid from the aminoacyl AMP and form aminoacyl tRNA. And here AMP is released and also charged tRNA with corresponding amino acid also released. So this process of joining tRNA with specific amino acid is called tRNA charging. Here, the use of phosphorylation or ATP is transfer of high energy bond to the tRNA molecule. And this stored energy will finally used for the formation of peptide bond during the translation. So this is the actual bonding, that is tRNA at the amino acid acceptor arm, that is there is a adenine residue and this is the amino acid. As you can see, amino acid is joined to the tRNA through a ester linkage to either the second position or the third hydroxyl group of the adenine residue. So this amino acid joined to the tRNA through the ester linkage to either the second position or the third position hydroxyl group of the third position adenosine residue. So this is all about the activation of amino acid and about the charged tRNA and also about the enzyme aminoacyl tRNA synthetase. Thank you.